DNA replication is a process by which the parent DNA copies itself to produce two identical daughter molecules of DNA. It occurs in between two cell division, the S phase and the interphase. Proteins are involved in DNA replication. Some of those proteins are the DNA polymerase, the helicase, the topoisomerase, DNA primase, single-strand binding protein, and DNA ligase. There are three steps that occur in DNA replication. One of the steps is the initiation, the elongation, and termination. Let's look at DNA replication in prokaryotes. A site called origin of replication consists of adenine and thymine rich base pair, serving as a starting point for replication. DNAA binds to the origin of replication, forming DNAA to DNA complex. The binding of the DNAA to DNA recruits DNAA. The DNAA is also known as helicase, causing processive unwinding of the DNA. The helicase also breaks hydrogen bonds between base pair, forming replication purpose, spreading bidirectional from 5' end to 3' end. The unwinding process requires ATP hydrolysis. ATP hydrolysis may cause topological stress, which leads to positive supercoiling. Type 2 topoisomerase, also known as DNA gyrase, relieve tension, allowing DNA relaxation and resealing. After which the single-strand binding proteins, also known as DNA helix destabilizing proteins, bind to the separated strands that was separated by helicase. Since it lacks enzymatic function, it keeps the two strands separated, providing a template for DNA synthesis and protecting single-stranded DNA from nuclease degradation. Other proteins like DNAC and DNAG assemble for initiating DNA synthesis. The second step, which is elongation. The two strands that are separated, one of it is called leading or continuous strand, and the other is called lagging or discontinuous strand. The template determine base sequence for the newly synthesized complementary DNA. DNA primase initiates RNA primer synthesis on lagging strand. Leading strand has a single RNA primer with an OH at its 3' end, which can be elongated with the addition of the deoxyribonucleotide, removing a phosphate in the presence of DNA polymerase 3. Lagging strand. In the lagging strand, the polymerase lack its activity there, or let me say the polymerase enzyme is not there to aid the addition of nucleotide in the lagging strand of the parent DNA. Okazaki fragments, which are the discontinuously synthesized DNA, are joined to form a continuous strand of DNA by two enzymes, which are the DNA polymerase 1 and DNA ligase. At the third step, which is termination, the recognition fork that was formed as a result of the parental DNA separation meets the terminus region having 20 to 24 base pair sequence called termination recognition. The termination recognition functions as a site termination utilization substance to bind to the termination recognition, can arrest a replication fork from one direction and stop the entire process.